Hello, everybody, and welcome to this discussion on gene cloning strategies. So before I delve into gene cloning strategies, let me discuss genomic sequencing in a bit. So genomic sequencing is occurring at a rapid rate. However, single gene cloning is still essential, and it is still essential because many genomes are yet to be mapped or sequenced, so you still need single gene cloning. And also, genome sequences only reveal partial information, in contrast to cDNA sequences, for example, which provide an array of information that I'll discuss in a bit. So when we say that many genomes are yet to be mapped or sequenced, you have to keep in mind that in such a case, one is ought to work at the single gene level. And also, we're interested in genomic sequencing, but genome sequences only provide partial information. So this is in contrast to cDNA sequences. cDNA sequences can provide us with expression profile information on differing cells at various developmental stages. They can provide us with expression profiles in response to natural stimuli, and they can also provide us with expression profiles in response to experimental stimuli. And so cDNA sequences are sequences that are reverse transcribed from messenger RNA, as you likely know. And it offers further information on the functional aspects of genes, such as expression profiles and biochemical functions, keeping in mind that the functional annotation of genomes always lags behind the structural annotation of genomes. And this makes sense. You have to know the structure in order to get to the function of a particular annotation. And this translates into the necessity of single gene cloning. In addition, in higher organisms, cDNA sequences offer information on splice isoforms along with the respective abundance of those isoforms. So you have to keep that in mind. Now, suppose that we wish to perform single gene cloning. What we have to do is we have to isolate a single gene from a complex source. So say this complex source is genomic DNA, for example, or cDNA. And so let me discuss isolation of a single gene from a complex source. So in terms of isolating a single gene from a complex source, one method to quickly run through a large numbers of unwanted sequences is needed. So there are two methods by which we can do that, by which we can isolate a single gene from a complex source and by which we can run through a large number of unwanted sequences. And so there are cell-based cloning strategies. These can be termed the library approach. And alternatively, there's the PCR approach, whereby we selectively amplify target sequences with PCR. And so in the cell-based cloning strategy or the library approach, source DNA is divided into manageable fragments, and then we clone everything. And so in this approach, let me discuss it in further detail. In the cell-based cloning strategy or the library approach, the source DNA is divided into manageable fragments, and then we clone everything. And then the collection of which would be called a library, and after which the library is screened to identify the clone of interest. Alternatively, the PCR approach is whereby we clone an individual fragment after selectively amplifying the sequence with PCR, the target sequence with PCR. So when I say that we want to isolate a single gene from a complex source could be genomic DNA or cDNA. So for example, consider the human genome. If you want to isolate a single gene from a human genome, that's a lot of unwanted sequences that you have to get through. And that's a lot of unwanted sequences that you have to run through. And so this can be done, for instance, from a genomic library or a cDNA library. These are the complex sources by which you have to quickly run through a large number of unwanted sequences. So a genomic library is generated by fragmenting the genome first, after which, so we generate a genomic library by fragmenting the genome first and followed by cloning overlapping fragments into vectors. The result of this is a genomic library, yet several problems arise with this approach. I'll discuss them in a bit, but they include that the gene may be cut internally, which is likely the case if the gene is large. And so you're fragmenting, and let's say the gene is right here and you fragment the gene. And so another problem that can happen is that you can have fragments that are small and you want to obtain a cushion of flanking regions around the gene. And so the problem with this is that if you don't have cushions around the gene, then this can cause problems. What we ideally want is, first of all, that the gene is not cleaved internally, but also we want a cushion around the gene when we're generating a genomic library. So for example, we could fragment the genome with EcoRI, for example, and on average, we'll get 4 kilobase fragments. And then we can insert the overlapping fragments into vectors such as phage lambda vector. But the problem with the EcoRI digestion is on average, it'll provide us with 4 kilobase fragments. And this is too small. This can be small. And instead, it's desirable that we obtain a cushion of flanking regions around the gene. And also, there is the potentiality with smaller fragments. 4 kilobases is not that small, but there is a potentiality that the gene may be cleaved internally, especially the case if the gene is large. So we can overcome these problems by cloning random DNA fragments of large size, say 20 kilobases instead, and then use lambda replacement vectors as the vectors for cloning overlapping fragments within those lambda replacement vectors. So the larger sizes translates into the following. So the larger sizes of fragments, DNA fragments, translates into fewer clones required for completing a library. So you have fewer clones that are required to complete the library, but also you obtain a large cushion of flanking regions around the genes, which is desirable. And then also the fact that it's a random fragments, if you've noticed, means that you have no systematic exclusion of sequences. And here, it doesn't mean that there's no exclusion of sequences, but it just means that there's no systematic exclusion of sequences. And so there's also the advantage of that. Now, we can calculate the number of independent recombinants required to achieve a certain probability of including a sequence in a library. And this is through this equation here, whereby capital N refers to the number of independent recombinants, P right here, so it's a natural logarithm of 1 minus P divided by the natural logarithm of 1 minus 1 over N, or a little case n. And so what you have to recognize here is the capital case N here refers to the number of independent recombinants. P refers to the probability of including any particular sequence in a random library. And little case n refers to the size of the genome relative to a single clone gene. And so the size of the genome relative to a single clone gene is something to keep in mind. And so we have to ask ourselves, what is the appropriately sized fragments that we want to obtain? How do we obtain those appropriately sized fragments? And so to produce appropriately sized fragments, we can use the Manieta strategy, which utilizes a mixture of two unrelated restriction endonucleases to perform a partial restriction digest. And this partial restriction digest produces large fragments, say 10 to 30 kilobases long. And then we can insert them into lambda replacement vectors. Now, these large fragments that are 10 to 30 kilobases long result in a random set of overlapping fragments. So the result of partial restriction digest is that, first of all, you get large fragments. Also, you get a random set of overlapping fragments. In addition, the mixture of two unrelated restriction endonucleases that are utilized are used usually such that you have tetranucleotide recognition sequences by those restriction endonucleases. So the restriction endonucleases have tetranucleotide recognition sequences. This leads to large fragments, and they also produce blunt ends. Another approach to produce appropriately sized fragments is to utilize single restriction endonuclease method. And the single restriction endonuclease method utilizes one restriction endonuclease that cleaves frequently. And so this is also used to perform a partial restriction digest. So also in the Manieta strategy, we performed a partial restriction digest, except we used a mixture of two unrelated restriction endonucleases, What you have to keep in mind here is that then the fragments are inserted into lambda replacement vectors, similarly to what we did in the Manieta strategy. So this is the single restriction endonuclease method and the Manieta strategy to produce appropriately sized fragments for your cloning. So an example of one singular restriction endonuclease that can be used for the single restriction endonuclease method is the SAW3A1. And the SAW3A1 can be used to cleave the DNA. And then when it comes to insertion into lambda replacement vectors, we can use the EMBL3 lambda replacement vector digested by BAMHI or BAMH1. What you have to keep in mind is that BAMH1 and the SAW3A1 both result in the same cohesive ends, so they result in the same sticky ends. And in terms of the EMBL lambda series of vectors, they have been widely used for the purpose of genomic library construction, and they facilitate selection based on the SPE phenotype, keeping in mind that the EMBL series of vectors contain the red and the GAM genes for the SPE phenotype selection. I won't discuss that here, but just keep in mind that they allow for selection based on the SPE phenotype because those vectors have the red and the GAM genes. So just to keep you oriented, so far I'm discussing genomic libraries, and I'll be delving into cDNA libraries in a bit, but let me continue with our discussion of genomic libraries. So we said that for the Manieta strategy and the single restriction endonuclease methods, we can insert into lambda replacement vectors for both. Yet different vectors can also be utilized in the case of genomic library construction. So we can use high capacity vectors such as cosmids, YACs, BACs, or PACs. There are disadvantages. There are difficulties in preparation of some of those high capacity vectors. So you have to keep in mind that there's difficulty or increased difficulty in preparation of those high capacity vectors such as cosmids, YACs, BACs, and PACs. What you have to keep in mind is that YACs, BACs, and PACs libraries have their main applications in genomic mapping and genome sequencing. In addition, however, there are advantages of high-capacity vectors, so you get larger average inserts, and this yields to increased likelihood of genes being included in a single clone. I've discussed this earlier, but also a decreased number of clones required to assemble a contig.